Hello, everyone. Welcome to my spatial data analysis video tutorials. Today, I'm going to show you how to analyze the dataset generated by the 10x Genomics Xenium in situ platform. So the dataset that I'm using for today's demonstration is the one-day-old mouse pup Xenium dataset. So we can go to 10x Genomics website and have a look at the dataset. So let's go to 10x Genomics website. So if you click the resources, then dataset, then you scroll down to the 10x instrument, you can click the plus button. Then you can see the dataset generated by different instruments. So we are going to use the dataset generated by the Xenium analyzer. You can see there are datasets available for you to download. So we can tick this box. Now all 17 Xenium datasets are shown here. So I'm going to use the whole mouse pup preview dataset. We can click this dataset. So you can see this dataset was generated by the Xenium onboard analysis 1.60 version. Down here are the information for the dataset. You can see the marker genes for different organs: brain, kidney, pancreas, liver, lung, heart, skin, colon, and thymus. Here is the material and tissue preparation information. And also importantly, you can see here the gene payloads. In this dataset, 379 gene probes were used to detect the gene expression on the tissue. If you click this link, you will download an Excel dataset to show the gene information. I downloaded already. We can go to my folder and have a look at the gene information. You can see this is the Xenium multiple V1 metadata. This is the gene information for the mouse dataset. We can open this Excel file. So you can see the gene information in the column A under the ensemble ID, the number of probes for each gene that were used for in situ hybridization. And importantly, you can see the cells and the tissue that express this gene. So here we have 379 genes. So for today's demonstration, I'm not going to annotate the cells, but if you want to see the cell types, you can use the information from here to see the gene expression in different tissue and cell types. So in the top, we can see the gene expressed in adipose tissue, then bladder, bone marrow. If you scroll down, you can see the expression in brain, heart, eye, then kidney, intestine, limb muscles, liver, lung, lymph node, mammary gland, pancreas, skin of the body, thymus, tongue, and the trachea. So you know where you can find the gene information, and later when we create a Seurat object, we also have the 379 gene in the Seurat object. So let's go back to the 10x Genomics website. So if you scroll down further, you can see the tissue information. The most important is the dataset you can download from here. I'm going to use the full output dataset. You can see it is 63.8 GB. So it is a very large dataset. It contains 1.3 million cells. It will take time to analyze this dataset. But if you want, you can also download the subset dataset. This will be much quicker. You can use the code I will post in the comments to analyze the subset dataset. If you click this link, you will download the dataset. I downloaded already. So here is the dataset I downloaded from the 10 times Genomics website. When you first download it, it will be a zip file. You need to unzip the dataset. Then we can have a look at the data set inside the files. So you can see what the data set inside here. Because we are going to use Seurat to analyze the data set, we need the cell feature matrix data. In most data set, when you download it, the cell feature matrix data also is a compressed file. You need to unzip twice to get this folder, cell feature matrix. We can click, have a look the data inside this folder. You can see this folder contains the cell barcode, features, and the matrix data. So now the data is ready to load into R for the analysis. So we can go to R and perform the analysis. So for today's analysis, we need the Seurat package tidyverse, and also we need the BP cell package. Because we have 1.3 million cells here, my computer will not be able to analyze the dataset using the standard Seurat workflow. Previously, I have a video to show you how to use the BP cell and analyze large dataset. I'm not going into details for how to use the BP cells today. If you want to know more details, you can watch that video. So I showed you how to download the dataset. Now we can load the dataset. Seurat v5 version has a function called node Xenium to load the dataset generated by 10 times Genomics Xenium analyzer. So we can just tell the files directory where we save the dataset. Under the field of view argument, we can set the field of view, or you can set a name you like. Then the C will be Xenium. Now we can run this code to load the data. Let's run. So I practice the analysis before today's demonstration. It will take at least 1 hour to load the dataset. So I will let the computer to run and come back 1 hour later to continue the demonstration. Okay, so we loaded the data. So next we can remove the cells from the data that have the 0 count. We can use the subset function to remove the cells. So we get rid of the cells that have the 0 counts. Next, we can use the wiring plot to look at the n features and n counts for each cell. Because we set the array as Xenium, now the n features will be n features Xenium and the n count will be n count Xenium. So let's run the wiring plot. You can see we generated the wiring plot for the n features and the n count. Because the in situ hybridization only used 379 gene probes, you can see the n features for majority of cells is below 100 and the n count is below 500. Because we also loaded the mouse whole body tissue section, so before we perform the cell clustering, we can use the image plot function to have a look at the gene expression on the tissue. I showed you on the 10 times Genomics website we can use some organ-specific mark genes to see their expression, and also we can identify their organs using organ-specific marker genes. So the GAD1 is a neuron-specific marker. MFAP4 is highly expressed in the lung. Then CLEC4F is expressed on the liver. LRP2 is a marker gene for kidney, and CDHR5 enable the colon. So let's run the image dim plot with those marker genes. We can run because it is a large data set. It takes time to show the image. Okay, you can see we generated the image. Let's zoom in, then we can see it more clear. So now you can see the whole sections of the mouse pulp tissue. We used some organ-specific marker genes, but you can see clearly from the image, you can see the structure. You can see here is the nose, the mouth, and the eye, the brain. Because GAD1 is a neuron-specific mark genes, you can see GAD1 is highly expressed in the brain. Then you can see the fornix. Here is the lung. The lung express high level MFAP4. Then here is the liver. We use the CLEC4F mark gene to show cells in the liver. Then here is the kidney, and the kidney express high level ERRP2. Then CDHR5 labels the colon. You can also see the tail of the mouse. So you know we have 379 genes there. You can just plot any gene that you are interested in. So next we can use the image feature plot function to visualize the expression level of genes at the per cell level. For example, we can use the AGER. It is the mark gene for the lung type 1 epithelial cells. We can run. Unlike the single-cell RNA sequencing dataset, you have lots of genes available to plot. But here you have to use the 379 genes. You cannot use other genes to plot the images. Okay, we generated the image again. You can see we show the AGER expression in red. Then you can see AGER is expressed in the lung. We can also zoom in to see the images. You can see the red color on the lung tissue. So at this stage, you can save the Seurat object. Next time when you need it, you can just load the Seurat object. I showed you when we load the original dataset, it almost takes 1 hour to load the data. But if you save the Seurat object here, next time when you want to load it, it just takes 1 or 2 minutes, depends on your computer memory. So I saved the Seurat object already. I don't need to save it again. So it is very convenient if you save the Seurat object here. Then you can stop from here for the analysis. Next time when you want to continue the analysis, you just need to read the Seurat object back into R again. So let's try to read the Seurat object back into R. We can run. So this time should be very quick. It only takes 1 to 2 minutes. We can reload the dataset. You can see now, this time we can quickly load the Seurat object back into R. So now we can perform cell clustering for the Seurat object. So if you have a small dataset, you can run the single cell transform data normalization, then followed by standard Seurat workflow analysis. Because I downloaded the full dataset, we have 1.3 million cells in the full dataset. My computer will not be able to analyze the full dataset using the standard Seurat workflow analysis. So I'm going to perform a sketch-based analysis for the full dataset. So before we perform the sketch-based analysis, we need to normalize the data. Let's run the data normalization. So after the data normalization, we identify the variable features. So normally we use the function find the variable features to identify variable features for downstream analysis. If we perform single-cell RNA sequencing data analysis with the find variable features function, we normally identify 2,000 genes as variable features for downstream analysis. But today, in this dataset, we only have 379 genes. So we are not going to run this function. Instead, we can just set all the genes as variable features. So we know all the genes are in the row names of the Seurat object. So we can set the genes as variable features. Before we set it, we can have a look at the Seurat object. So let's click the Xenium object. You can see in the Cs, we have the Xenium assay. Inside the Xenium assay, you can see at the moment the variable features is 0. So let's set all the genes 379 as variable features we can run. Let's have a look at the Seurat object again. You can see now the variable features is 379. So we set all the genes as variable features. Now we can perform the sketch-based analysis. Before we do that, we can set the future globals option, the maximum size as 10 to 9. Then we set the Seurat version as V5 because we use the Seurat V5 version to perform sketch-based analysis. Let's set it as V5. Now we can sketch the data. I'm going to sketch 50,000 cells from the 1.3 million total cells. So the method will be average score. We will use this score to project to the full dataset, and the sketched array will be sketch. So let's run. You can see Seurat is calculating the average score for each cell. After Seurat finishes the calculating average score, it will create a sketched array in the arrays. Then we can have a look later for the sketched array. So Seurat finished the calculating average score. Because we just sketched the data, the default array should be sketched. You can use the default array to look at the Xenium object. If we run, you can see it is sketched. If you change the default array, you can set as sketched using this code. So now we can run a standard Seurat workflow for the sketched data. First, we scale the data. Now we only scale the data for the 50,000 cells. So next we can run PCA. Because we set the features as the row names, so the features will be the 379 genes. You can also set the features here as row names. So after PCA, we run UMap. After UMap, we can find the neighbors. Now we can find the clusters. I'm using the resolution 0.5. If you look at the data on the 10 times Genomics website, they identified more than 60 clusters because they use high resolution. I tried this dataset. If I set the resolution as 2, then I can identify 67 cell clusters. Here, because I set as 0.5, you can see we identified 27 cell clusters. So now we can use the plot function to look at the cell clusters from the sketched array. We can run. You can see we have a UMap here. It is the sketched array. It contains 50,000 cells. And we have 27 cell clusters here. Now we can run the feature plot for organ-specific genes that we ran in the beginning. GAD1 is the mark for neurons, AGER is the mark for lung type 1 epithelial cells, CLEC4F is the mark for the liver, and LRP2 is the mark for kidney cells. Let's run the feature plot. We can zoom in. You can see GAD1 label this cell cluster, so cells in this cell cluster should be neurons. AGER label this cell cluster, so cells in these clusters are cells from the lung, and this cluster should be cells from liver, and here are the cells from the kidney. You can see the sketched array can cluster cells into distinct cell clusters according to their location in different organs in the mouthpulp. So let's have a look at the metadata for the Xenium object. Now we can run. So you can see in the metadata we have the uncount Xenium, unfeatured Xenium, and importantly at the end we have the average score. When we perform the sketch data, so Seurat calculated the average score for each cell. Next, the sketched analysis for the 50,000 cells. NA means no cells are not in the sketched dataset. For example, this one is in the sketched dataset. It is in the cluster 2. Then this column is the Seurat clusters. So now we have the cell clusters. We can use the image plot function again to look at all the cell clusters on the tissue. We can run the image plot. You can see we have a warning message here, no field of view associated with sketched array. Seurat uses the global default field of view images. Now we can zoom in to see the cell clusters on the image. You can see we have 27 cell clusters here. From the color for each cell cluster, you can clearly see cells from different organs have been clustered into distinct cell clusters. You can see cells from the brain, they are in green color, and cells in the lung, they are in blue color. And also you can see liver, kidney, skin cells. So we only sketched 50,000 cells from the 1.3 million cells, but you can see the analysis clearly show organ-specific cell clusters. We can also plot some organ-specific mark genes together with the cell clusters. We are going to use the GAD1 for neurons, MFAP4 for the lung, CLEC4F for the liver, then LRP2 is expressed on the kidney, and the CDHR5 is expressed in the column. Let's run the image dimplot function again. And we can zoom in again. You can see now we not only show the 27 cell canisters on the tissue, we also show 5 organ-specific genes expression on the tissue. So you can see, use the sketched assay, we can cluster the cells based on their location on the tissue, and the analysis worked really well. And the next image is very cool, I think. So now we analyze the 50,000 cells. The 50,000 cells can represent the whole data set. You can just use the sketched assay to perform your analysis because the cells from the sketched data should represent the whole data set. If we look at the Seurat object now in the assay, you can see we have a sketched assay here. But we can still project the sketched assay to the full cell dataset. Then we can analyze the full dataset. Because the full dataset has 1.3 million cells, so everything we are going to analyze on the full dataset will be very slow. So to analyze the full dataset, we can project the sketched data on the full dataset. So let's project the sketched data on the full dataset now. Once again, it will take some time to project the data. Okay, you can see we successfully project the sketched data analysis on the full dataset. So now we can set the default assay as Xenium to analyze the full dataset. We can run, and also we can have a look at the metadata now. And we look at the encounter features, we can go to the end. You can see now, for all the cells in the full dataset, they have been allocated into one of the clusters. And here is the cluster full score. So now we can go back to R and use the dim plot to look at the full cell clusters. Let's run. As I mentioned, now we have 1.3 million cells. Everything we run will be very slow. So you can see we generated the UMAP for the 1.3 million cells, and here we still have 27 cell clusters. Again, we can use the feature plot to look at the gene expression on the cell clusters. For the demonstration, I'm going to use the AGER again, because it is a non-type 1 epithelial cell specific mark gene. Let's run. So we know from the sketched assay, non-cells are in cluster 5. Now we should still see AGER expression in cluster 5 in the full dataset. So you can see in the full dataset, AGER still labels the cluster 5. So we just use AGER as a demonstration because I know well the gene expression in the human and the mouse lung. So if you work on different organs, you can use the tissue-specific genes in your research area. So finally, we can look at all the cells on the tissue. We can run the image dim plot function again. Under the group by argument, we will use the full cell clusters. Let's run. Okay, we generated the image. Let's zoom in to see all the cells on the mouse tissue. You can see we have 27 cell cluster here, and we analyzed the 1.3 million cells from the whole mouse tissue. So now you can save the Seurat object Next time you want to perform other analysis, you can just reload the dataset. You don't need to run the analysis again because the whole process is very slow. Okay, I'm going to stop from here for today's demonstration. In summary, I showed you how to analyze spatial dataset generated by the 10 times Genomics Xenium platform. And also, I showed you how to use sketch-based C to analyze large dataset. So you can download the data and practice the method. And I hope my video tutorial can help your data analysis. Again, please subscribe my channel and share my videos with your friends. Thank you. See you next time.